In this video, we'll discuss about purine nucleotide synthesis or de novo synthesis of purine nucleotides. De novo synthesis of purine nucleotide. Sources of carbon and nitrogen in purine ring. It is purine structure. It numbered in a anti-clockwise direction. There is double bond. This one is the purine structure. In this purine structure, C4, C5 and N7, it is from glycine. N3 and N9, it is from amide nitrogen of glutamine. Amide nitrogen of glutamine. N1 from aspartate. C6 from carbon dioxide. C8 from tetrahydrofolate, that is N5, N10 tetrahydrofolate. C2 from tetrahydrofolate, formyl tetrahydrofolate. So these are the sources of carbon and nitrogen in purine ring. C4, C5, N7 from glycine, N3, N9 from amide nitrogen of glutamine, N1 from aspartate, C6 from carbon dioxide, C2 from formyl tetrahydrofolate, C8 from N5, N10 methylene tetrahydrofolate. These are the sources of carbon and nitrogen in purine ring. Coming to de novo synthesis of purine nucleotide, there are three main sources or three main pathways synthesis of purine in our body. First one is de novo synthesis. Second one is purine salvage pathway. Third one is phosphorylation of purine nucleoside. So first one is de novo synthesis. Second one is purine salvage pathway. Third one is phosphorylation of purine nucleoside. Of this, de novo synthesis is the predominant pathway. First one is the predominant pathway. Purine are dietary non-essential. They are synthesized from amphiphilic intermediate, that is known as de novo synthesis. That is synthesis of purine from amphiphilic intermediate. It is known as de novo synthesis of purine. This is purine nucleotide synthesis. There is synthesis of IMP first. So purine nucleotide synthesis, first step IMP first. So from the free exciting ribose 5-phosphate, first step ribose 5-phosphate, purine are built up on free exciting ribose 5-phosphate. There is synthesis of IMP. First, from IMP will get GMP and AMP. These monophosphate will converted into diphosphate, guanosine diphosphate and adenosine diphosphate. This diphosphate is converted into triphosphate, guanosine triphosphate and adenosine triphosphate. There is also diphosphate conversion of ribonucleotide into deoxyribonucleotide. So this is overall synthesis. So ribose 5-phosphate with having 11 steps. After that it will converted into IMP. This IMP finally converted into GTP, ATP. So this is the overall synthesis. Now coming to synthesis of IMP, there are totally 11 steps in the synthesis of IMP. In this first, third, fifth, sixth, eighth steps required ATP as well as they catalyzed by synthetase. So first 3, 5th, 6th, 8th steps required ATP as well as synthetase enzyme. Step number 2 and 5 required glutamine. Then step number 3 required glycine. Fourth and 10th step required tetrahydrofolate. Seventh step required carbon dioxide. Eighth step required aspartate. Ninth and 11th step not required anything. So these are the 11 steps. 1, 3, 5, 6 and 8 required ATP as well as synthetase enzyme. Second and fifth step required glutamine. Third step required glycine. Fourth and 10th step required tetrahydrofolate. Seventh one required carbon dioxide. Eighth step required aspartate. Ninth and 11th step not required anything. Now coming to IMP synthesis. The free exciting molecules for purine nucleotide synthesis is ribose 5-phosphate. First precursor molecules are ribose 5-phosphate which are synthesized in the HMP stunt. First reaction there is utilization of ATP and this ribose 5-phosphate is converted into phosphoribosyl pyrophosphate PRPP, phosphoribosyl pyrophosphate. This is the second step. Now this PRPP required glutamine and converted into 5-phosphoribosylamine. It required enzyme glutamyl amino transferase. Now third step 5-phosphoribosylamine required glycine and ATP. It converted into glycinamide ribosyl 5-phosphate. It required ATP and synthetase. So 5-phosphoribosylamine, presence of ATP, glycine and synthetase enzyme converted into glycinamide ribosyl 5-phosphate. It is the third step. Now this glycinamide ribosyl 5-phosphate is converted into formyl glycinamide ribosyl 5-phosphate. It required enzyme transferase. Then it also required N5-N10 methynyl tetrahydrofolate. Now this formyl glycinamide ribosyl 5-phosphate required ATP and synthetase glutamine. It converted into formyl glycinamide ribosyl 5-phosphate. It is the fifth step. After this formyl glycinamide ribosyl 5-phosphate acted upon synthetase enzyme and ATP utilization. It will converted into amino imidazole ribosyl 5-phosphate. Amino imidazole ribose 5-phosphate. It is the sixth step. Amino imidazole ribosyl 5-phosphate with the help of carboxylase enzyme and carbon dioxide converted into amino imidazole carboxylate ribosyl 5-phosphate. This is the seventh step. After that it is acted upon this amino imidazole carboxylate ribosyl 5-phosphate utilization of ATP and aspartate converted into amino imidazole succinyl carboxynamide ribosyl 5-phosphate. Then this one acted upon enzyme lyase and fumalate. It will converted into amino imidazole carboxamide ribosyl 5-phosphate. Then this one will acted upon enzyme transferase. It required formyl tetrahydrofolate converted into amino imidazole carbosamide ribose 5-phosphate. For aminoimidazole carboxynamide ribose 5-phosphate. This one acted upon enzyme cyclase removal of water molecule converted into IMP. Iodine monophosphate. IMP. This one is the last 11th step. Iodine monophosphate. This glutamyl amino transferase, it is a regulatory steps. It is a regulatory enzyme also. This glutamyl amino transferase, this PRPP synthetase, phosphoribosyl pyrophosphate synthetase, 2 are regulatory enzymes. Now this IMP is converted into adenylyl succinate and xanthosine monophosphate. This IMP is converted into adenyl succinate. This adenyl succinate is converted into AMP. This adenyl succinate is converted into AMP. It is required aspartate and GTP. It required in this process. It required aspartate and GTP. This IMP is converted into xanthosine monophosphate. This xanthosine monophosphate is converted into guanosine monophosphate. It is NAD dependent reaction. This IMP dehydrogenase reaction. This one is the IMP dehydrogenase reaction. With the help of that glutamine, ATP and synthetase, it will converted into GMP. There is cross regulation. GMP required ATP, AMP required GTP. So it is a cross regulation. If one nucleotide is deficient, there is deficiency of other nucleotide. This is important of cross regulation. Also, this enzyme adenyl succinate synthetase and IMP dehydrogenase, these two enzymes, these are under feedback control AMP and GMP. This monophosphate synthetase, nucleoside monophosphate, adenosine monophosphate and guanosine monophosphate, they converted into nucleoside diphosphate. This one acted upon enzyme kinase converted into NTP, nucleotide triphosphate. That means ATP and GTP. 11-step reaction, IMP is converted into AMP and GMP. By the kinase, it converted into nucleoside triphosphate. NTP is converted into deoxynucleotide diphosphate. With the help of nucleotide reductase, it required thioredoxin and NADPH. Thanks for watching.